It's Amy Bowles' 9th video, and I've broken my Pioreactor. That's not the worst of it though. We have a 50% chance of exceeding 1.5 degrees of global warming in just 7 years' time. As crop failures become more common, things like solar-powered bioreactors could become a life support system. On the bright side, in the last 2 months, right up to my breaking the Pioreactor, we've been making some pretty immense progress on the research that we can do at home into developing single-celled protein. We started out by finding an affordable open-source bioreactor. We initially trialled it with some spirulina. The Pioreactor stirred the culture, illuminated it with a pair of white LEDs, measured and maintained its temperature, and measured its optical density. Then I installed an Adafruit spectrometer to measure its colour. I'll be analysing that data over Christmas. We then added a 50ml centrifuge tube, filled it with culture media, and hooked it up to a peristaltic pump. This would mean that we could operate in fed batch mode, adding nutrients at appropriate times. But we also added an empty tube and second peristaltic, so we can theoretically avoid overflow, harvesting culture while replenishing nutrients and working in chemically static or chemostat mode. And better still, by using optical density measurement, the Pioreactor can keep the turbidity, or the concentration of cells, constant. This is called a turbidostat. I aim to report back on the turbidostat and spectrometry results next week, so subscribe if you'd like to see those. Then I'm hoping to have the Pioreactor fixed in time to start a new culture in 2 weeks' time. Spirulina does let you very easily produce protein from nothing much more than sunlight and air. Single-cell protein, or edible unicellular microbes, are the most efficient way of producing protein because we eat the whole organism. So there is very little waste. Precision fermentation is brilliant because you can make any sort of chemical you want, like casein, so we could have a real vegan cheese. But we generally need to get rid of the biomass because it's just the casein that we want to eat, not the whole bacteria. With plants like soya, which are the most efficient plant to produce protein, I understand, it's worse because we only eat the beans. And with livestock, it's way worse because they only eat the beans, or grass, which takes up even more land, and we only eat bits of the livestock. But unfortunately, spirulina probably can't be the whole answer. It's a cyanobacteria which can produce cyanotoxins like microcystin, hence recommended daily doses of say 3 grams. If spirulina contained as much as 70% protein, then we're only looking at under 4.5% of a 60 kilogram adult's protein requirement. But every little helps, and they do have many other benefits. Hydrogen oxidizing bacteria also take carbon dioxide as their carbon source, but they can't use sunlight directly as their energy source, which some might say is a good thing because it means they aren't green and they don't taste green. Instead of photosynthesis, they can use hydrogen as their main energy source, which we can produce quite efficiently from sunlight via solar-powered electrolysis. So we could produce hydrogen oxidizing bacteria at home as a tasty single-cell protein with a Pioreactor and a tank of hydrogen and carbon dioxide. But hydrogen is famously explosive, so rather than using hydrogen gas, we tried to produce our own in a Pioreactor vial. And to my delight, it worked, and I didn't electrocute myself or even short out the power mid-live stream. You can see a few issues here though, the least of which was that the power supply was way bigger and heavier than the entire Pioreactor turbidostat. Much more importantly, my attempt at an electrode holder left a lot to be desired, and worse still, the water turned green as we dissolved one of the electrodes. Fortunately, within 2 weeks we managed to resolve all 3 of these issues. Cam proposed a neat self-contained solution that can be entirely controlled by the Pioreactor interface and only requires a single power socket. Rob designed some new caps that let us much better align the electrodes, and Narcissus pointed us to a platinum-plated titanium mesh that jewellers use to electroplate jewellery. I sawed off a strip of the £9.39 mesh, and Narcissus explained that the cut titanium wouldn't be a problem, as it would oxidise titanium dioxide, leaving the platinum coating as the conductive surface on the anode. Given that there was room for 10 of these, each platinum anode should cost around 94 pence. So, last week Garrett and I put them all together in this very neat automatic electroturbidostat, and it seemed to work very well for electrolysis. But it wouldn't work for continuous hydrogen oxidizing bacteria production. The problem is the oxygen. When we turn on the current, an oversimplified way of thinking about it is that for each water molecule we break, we get a diatomic hydrogen molecule forming on the cathode and half a diatomic oxygen on the anode. So breaking 2 water molecules gives us 2 hydrogen molecules on the cathode and 1 oxygen molecule on the anode. So as we maintain the current, we keep producing twice as much hydrogen as oxygen. The trouble is that the hydrogen oxidizing bacteria consume 4 hydrogens for every oxygen they need. So excess oxygen starts to build up on the electrobioreactor. When the oxygen reaches the cathode, it takes electrons from the cathode and hydrogens from around it and forms water. This cuts off the supply of hydrogen to the bugs, so they stop growing. So how did Narcissus get around this? He purged the solution with carbon dioxide to flush away the oxygen. I must confess that replicating this hasn't been as plain sailing as I had anticipated. We chose SodaStream cylinders as our carbon dioxide source, as they are common household objects, and more importantly, the empties can be swapped for full cylinders at various local supermarkets, so the environmental impact isn't too bad. We added a converter to take it up to standard cylinder size, then a regulator with built-in solenoid valve, bubble counter and check valve. This solenoid happens to come with exactly the right power adapter to plug straight into the Pioreactor hat. The first problem I had was backflow though. I came back the next morning to find the contents of my Pioreactor in the bubble counter. I think the issue is that I'd bodged a barbed lure connector to attach our 16th inch tubing to the 3/16 inch barb connector. This meant that when the solenoid closed, carbon dioxide leaked out of my non-airtight connection, so the pressure dropped and sucked up the culture. So, after discussing this in the forum, we bought some 4mm tubing and a 3/16 inch lure barb that should fit in it, hopefully, along with an extra check valve to be doubly secure. I didn't mention the second problem in the forum, as it's much harder to solve. I'm an idiot. So, while trying to work out why we were getting backflow, instead of connecting the carbon dioxide to the submerged sparging tube, I connected it to the vent port, pressurising the headspace, spilling culture over my Pioreactor, and poaching my heater PCB. And many thanks to Cam, who sent a replacement out already, so I'm hoping we'll be back on track in a couple of weeks' time. But long term, we've got to find a more sustainable solution than bottled carbon dioxide. In Monday's Journal Club, we briefly discussed my idea of running some useful fermentation that produced sufficient carbon dioxide to purge the oxygen from the hydrogen oxidizing bacteria fermentation. But Narcissus pointed out that the first culture would likely migrate through the airline. So perhaps our endgame is a mixed culture with something that consumes the excess oxygen and produces carbon dioxide through aerobic respiration? Please do let us know your thoughts in the comments. How can we deal with this? We've come so far in 2 months that I'm sure we can crack it together. Catch you in the next one.